Hi everyone, it's a pleasure to be here and a huge opportunity. As Cora mentioned, I'm a clinician. I'm a MD in Chile, currently pursuing a PhD in the genetics of Parkinson's disease, granted by the Michael J. Fox Foundation through the Global Parkinson's Disease Genetics Program, that you may know as GP2 for short. And also, I wanted to present to you today the pilot program we developed at NIH during my sabbatical. I have no disclosure more than I love treating patients and I seek the answer for them. So maybe most of you don't know Chile. Well, Chile is a long, large, narrow Latin American country that is as wide as wide the US is from east to west coast. And maybe some of you know Chile because there were 33 miners trapped more than 10 years ago, 700 meters underground. And they were there more than 2 months, and they were rescued because of international collaboration. And why do you think I bring this story to you today? Because I want to make an analogy. This is the real capsule that extracted the miners in Chile. And as no one thought that these miners could get out of the mine with limited resources and also with the difficulties of finding them, well, they got out of the mine. In the same way, I want to get deep into the DNA and try to understand and study the genetic diseases that my patients suffer. They come to me and seek for help for Parkinson's, for ataxia, for other types of overlapping movement disorders that sometimes underlie a genetic diagnosis that most of the time we can't get there. So that's my motivation and my drive, to try to get the answer for them. So today, we are going to talk within the rare disease background. When I mean rare disease, I'm simplifying a bit the things because I'm trying to put in the broader spectrum of movement disorders that can be caused by a genetic etiology. So we will go from identifying the best DNA extraction method for countries that don't have the high quality technology that some countries have, to what I think could be the future or some real solution for this kind of patients. So as many of you have heard and have known, and it has been discussed here, well, the genetic disorders that can be caused in rare diseases, but a wide and complex kind of genetic variants, from single nucleotide variants, structural variants. All of them have been studied for years with short read, with limited success, because you can't study, as you know, all of them with one single test. I will want to go into the specific case of repeat expansion that are diseases very difficult to diagnose. The hit was in 1991 with the discovery of the frataxin gene, the Friedreich's ataxia. And from then on, it has been an exponential discovery of different repeat expansion disorders. But most of these have to be studied in a slow and costly way, because you have to study, there's no panel that includes all of them. And there have been new discoveries that have been very important. With this slide, I don't want to make you dizzy or to leave the auditory. It's just to show you how a clinician is presented when he or she has to solve a case of ataxia, for example. So it's very complex. I want to say it was my fellow question for my final exam, because it's thought as the most difficult trouble you have to solve. So when you have some of these cases, you begin trying to get from the family history the inheritance, if you suspect that it's autosomal dominant, recessive, etc. And then you go through a lot of steps, one gene at a time, to try to get to the diagnosis. And this complex process that gets more complex if the age of onset is earlier, so you have different algorithms according to age of onset. Well, these complex algorithms have a worldwide a success rate of 30 to 50%. Our center, in the last 4 years, sending samples all over other countries. In Chile, we don't perform that. We had a success rate of 23%, but all of this paid out of pocket from the patients. I wanted to highlight this study that was recently published in the New England Journal of Medicine. They were trying to identify the etiology of unsolved cases. These cases were from exome sequencing that were negative, and they performed whole genome with a short read. The increase in the diagnostic rate was low 8%. But what I wanted to highlight here is that many of these diseases were neurological, and more strikingly, this increase was mostly for European population. So other ancestries didn't increase a lot in the genetic yield. So I think, and I hope I haven't bored you with this introduction. I wanted to give you a context of the problem, to say it in a way, and also I wanted to explain what drives me to try to answer this question. I was a clinician working peacefully in Santiago de Chile, and I decided, okay, I have to do something. I have to help these people. I have tried 6 years to try to do something, to build something. There was no infrastructure, there was no knowledge, anything. So 3 years ago, I decided to join the Global Parkinson's Genetic Program as a PhD student to try to get to the genetics, in this case, of Parkinson's disease. But what my main objective was to move far beyond Parkinson's, because I see all kinds of patients. So in my mind was, when I have the opportunity, I will go and get it, and I will try to study these other cases. So the opportunity came, that's why I'm here, and I'm showing these pilot project objectives, which is, in simple, changing that algorithm that I show you, that was really complex, really expensive, and with a lot of steps, to translate that in one unique genetic test that solves all of them, that can identify all the underlying genetic etiologies. So this was my main objective, and also, well, identify the genetics of the undiagnosed cases. So our hypothesis was that long-read could solve this issue. So as part of my PhD, I went to NIH to do part of my sabbatical for my PhD project that is on genetics of Parkinson's. But I split my time to work with the long-read team sequencing team. So this picture that is taken at NIH, it's supposed to be a happy picture. But as you know, some pictures may deceive you or they don't show what they really are. So this picture is very important to me because when the samples from Chile arrived, they almost didn't have any DNA. So I have reached almost 300 patients with Parkinson's. I have collected the blood samples. I have extracted the DNA in a commercial lab. And when they arrived to NIH, they had less than 4 micrograms of total DNA. So I have to be honest with you, I cried. I cried that day. And I didn't know what to do because only 30% of the samples of my PhD project were able to go this short-read panel. And my dream of going into long-read was impossible because imagine, less than 4 nanograms of DNA, and it had to go to a gene panel. So I was really in a difficult situation. So what could I do? I thought, okay, I could try to do the same things that I was seeing that were done at NIH, these super fancy machines, robots, and everything to extract the DNA. I have to confess, I have used personal money to solve these issues, but I couldn't buy a machine a robot for extracting DNA. It was not possible. So, well, another possibility was okay, I can ship blood from Chile. But that wasn't a possibility because I have been seeing these cases for 2 years. So it was not possible to go 2 years back and take the samples and ship them. So it was really difficult. So it was then when we decided, okay, let's try to see different protocols for DNA extraction, different kits that could be performed in any place at a low cost, and that could be very easy to implement, both for the technology and from the people that did it. So here I have to give another disclosure, that is that I have never touched a pipette until January of this year. Never. Never, never, never touch a pipette. And I had to extract DNA. So it was like, okay, let's go. And after a lot of work, a lot of hours trying, and everyone was celebrating, I don't know, New Year and different things, I was in the lab trying to get to first get the pipette to work. So after this work, we got this protocol, this nice protocol that you can see if you take a snapshot of the QR. And you can see the huge difference. I began with 0 DNA to a protocol that gave me the huge amount of 50 micrograms of total DNA. This was a success. And with a nice average phenotypic of around 35 kb. So it was a tremendous success, but we didn't know how will the data will be generated, if we would have success or not for those steps. So we began working on the data analysis, but as you know, resources are limited. We couldn't run long read for all these cases, so we had to select based on the high probability of having a genetic disorder. So that is why we chose 15 samples that had family history or early onset, and also that have negative previous results. I went back to Chile. My sabbatical was 3 months. I extended 3 more months. I extracted all the blood that was in the fridge. I shipped it to NIH, and then they continued working on this. These samples were size selected. The 10 kb that were short read were removed, and then the library preparation was done manually. And they were loaded 1 flow cell per sample, and they were done 3 loads for 72 hours. And you can see here this cheap protocol done in Chile and shipped to the NIH gave a nice data output of almost 130 gigas with a coverage of more than 40x and with a Q-score of 23. Here we can also see that the N50s were around 23. That was quite nice. And also we tried to see if we could perform de novo assembly with these samples. But as you see this messy untangling thing, we couldn't perform de novo assembly because they were from short read. But anyway, we continued the analysis and we processed the data based on the different types of variants. We had a pipeline for structural variants, for single nucleotide variants, and for structural variants. Then you can see here, and for sake of time, I will not explain more in detail. So for the results, this was a very hard part because we started with the single nucleotide variants. That was more easy. We selected those variants related to Parkinson's disease or ataxia genes. They were 85 that we selected. And we then began filtering by minor allele frequency less than 1%, and then the ones that had high probability of being pathogenic, that had a CAD score more than 20, and then we manually scored them with the ACMG guidelines. Some of the variants we identified were in the common Parkinson's disease genes, GBA1, one of the main risk factors, LARC2, a common genetic variant, and some interesting findings on the SPG7 gene that we should continue exploring. Regarding short tandem repeats, this is the repeat expansion catalog genes that we looked for. We didn't identify any pathogenic variant in these samples. We did identify one expansion in the BIN1 locus, but the motif that is seen in orange, the TAAA, is not pathogenic. And for structural variants, in the first analysis, we didn't identify any pathogenic structural variants in these genes. Yesterday, I was very interested in Gus' talk about needle. They are just in this moment working in this at NIH. They are seeing us and working on that. We also try to look in another way to look through the annotation if there were some deletions that could be pathogenic, and we identified this deletion that is more than 100 kb of size. It is related with some neurological genes. It's very infrequent, but it's not directly related to Parkinson's or ataxia. There are some regulatory elements that could be related to the phenotype, but we need to go further in the analysis, like doing RNA-seq or some validation to know if this is pathogenic or not. I will skip that. So in summary, we developed a high-quality protocol for DNA extraction. We identified some single nucleotide variants in PD genes. We got to accurately size repeat expansions. And with future tools, we hope to do some follow-up analysis and try to look deeper into the data. Our future work includes, for me, finishing my PhD and defending the end of October this year. And for my patients and the research, well, we are trying to develop an adaptive sampling protocol for looking to all these regions, the repeat expansion regions, and also the point mutations for ataxia and Parkinson's genes. So this picture is from Madison Fulcher, an NIA summer intern, that this adaptive sampling in 15 cases, not my cases, other cases. And you can see here that the coverage was pretty good, overall 50K of coverage for ataxia repeat expansion. And when comparing a positive carrier of RFC1 with the whole genome sequencing, the standard long-read protocol versus the adaptive sampling, you can see that you get more reads and more homogeneous expansion with the adaptive sampling. So I would like to thank this amazing opportunity to Nanopore, to all the people that have helped me at NIH, the CAR team, especially Kimberly Billingsley, that I know she's online and couldn't be here.